All right, let's talk about influenza. You know, that virus that seems to trip everyone up on exams with its tricky mechanisms? Well, you're in the right place. Welcome, everyone, to a super high-yield explainer that's going to help you absolutely nail this topic for your exams.
Okay, so here's our game plan. We're going to dive right in and break this all down. We'll start with the basics, the family it belongs to, then we'll look at its structure, its really clever tricks to outsmart us, and finally, how it actually attacks the body.
First things first, let's get our bearings. Where does this virus fit in? Well, influenza belongs to the Orthomyxoviridae family. You got to remember that name, Orthomyxoviridae. It's a huge group of respiratory pathogens, and trust me, you'll be seeing them a lot.
So here's a super important point, and I guarantee you this is a classic exam question. That O in Orthomyxoviridae, think of it as standing for odd one out. Why? Because almost every other RNA virus replicates in the cytoplasm, right? Not influenza. This guy is different. It has a segmented RNA genome, and it replicates inside the host cell's nucleus. That is a golden high-yield fact. You absolutely have to lock that one in.
Okay, moving on. Now that we know where it lives, let's zoom in and dissect the virus itself. We're going to look at it piece by piece, kind of like looking under the hood to see what makes this engine run so effectively.
Check this out. This image gives us a perfect blueprint of the virus. You can see that segmented RNA core we just talked about, wrapped in its protein shell. But look closer. See those little spikes sticking out from the surface? Those are the glycoproteins, Hemagglutinin, and Neuraminidase. And believe me, these are the virus's main weapons. We're going to be talking a lot more about them.
So here's the arsenal, the virus's toolkit. For your exams, you really need to focus on two of these proteins. First up is hemagglutinin, or HA. This guy is shaped like a triangle, and his job is all about viral entry. So think HA for hello attachment. It's how the virus gets in. Then you have Neuraminidase, or Na. It's mushroom-shaped, and it's an enzyme that lets the virus get out. So think Na for need to get away, or ADS. HA and Na, these are the big targets for our immune system, and really importantly, for our antiviral drugs.
All right, now we get to the really clever part. This is the secret to influenza's success, its ability to constantly change its disguise. We're about to explore exactly how it keeps modifying its appearance, which is how it evades our immune system and keeps coming back to infect us year after year.
So have you ever wondered how the flu manages to come back every single year? How does it cause everything from that seasonal bug to a full-blown global pandemic? Well, the answer comes down to two super important processes, Antigenic Drift and Antigenic Shift. Getting the difference between these two, let me tell you, it's absolutely crucial for your exams.
Okay, this table breaks it all down for you. This is pure gold for your exams. Let's start with antigenic drift. This is a slow, gradual change. Think of it like tiny little typos or point mutations that happen in the HA and NA genes. It's subtle. This happens in both influenza A and B, and it's the reason we get seasonal flu and need a new vaccine every single year. Now, antigenic shift is a whole different ballgame. This is a major, abrupt, dramatic change. It's not a typo. It's like swapping whole chapters of a book. This happens when different influenza A viruses actually swap entire gene segments. This is what can lead to a massive pandemic because it creates a brand new virus our immune systems have literally never seen before.
And of course, we need a way to remember this. So here's a super simple mnemonic so you never mix these up again. For drift, just think deviating slowly with point mutations happening kind of daily. Simple, right? Now for shift, think sudden, severe, and it comes from reassortment. Got it? Drift is slow, shift is sudden. Easy. And this image? This shows you exactly how a big, scary antigenic shift can happen. Look, you've got a pig. We call it the mixing vessel, and you'll see why in a second. So this pig gets infected with a human flu virus and an avian or bird flu virus at the same time. Inside that pig's cells, those segmented genomes we talked about, well, they get all mixed up and shuffled around. It's like a genetic lottery. And what comes out is a brand new Frankenstein-style hybrid virus that could potentially spark a human pandemic.
Okay, so we know who the enemy is, and we know its master strategy of drift and shift. So now, let's head to the battlefield, the human body. We're going to trace the virus's plan of attack, step by step, from the moment it gets in to the damage it leaves behind.
So the infection process is pretty straightforward. It follows these 4 steps. Step 1, it gets in, usually through respiratory droplets. Step 2, it attaches. Step 3, it replicates. And Step 4, it spreads and causes a whole lot of damage. But the real key to understanding why some flu viruses infect birds and others infect us, it all comes down to Step 2. It's all about what specific receptor that HA protein attaches to.
So what are these sialic acid receptors? The best way to think about them is like locks on the surface of our cells in our respiratory tract. And that hemagglutinin protein on the virus, that's the key. Whether or not the virus can infect a cell depends entirely on whether its key fits that cell's specific lock.
Okay guys, this table is beyond critical. You have to know this. Look, there are 2 types of locks or receptors. Human flu viruses, they love the alpha-2/6 receptors. And guess where those are? All over our upper respiratory tract, our nose and throat. That's why seasonal flu gives you the classic sore throat and cough. It stays up high. Now, avian or bird flu viruses, they prefer the alpha-2/3 receptors. We don't have many of those up top, but we do have some deep down in our lungs. That explains 2 things. 1, why it's pretty rare for humans to get bird flu, but 2, why if we do get it, it can cause a really severe, deep pneumonia. It's hitting us where it hurts.
And boom, this brings it all together. This is why we call pigs the perfect mixing vessel. Remember that diagram? Well, it's because their respiratory cells have both types of receptors. They've got the alpha-2/3 and the alpha-2/6 locks. This means they can get infected by human flu and bird flu at the same time. This creates the perfect storm, the perfect environment for that genetic reassortment, that big, scary antigenic shift that can cook up a brand new pandemic strain.
So let's wrap this up. We've seen how this virus can constantly drift, making small changes every year. And we know it has the terrifying potential to shift, creating something completely new. So knowing all that, what do you think is the single most important public health measure we have to prepare for the next pandemic? The answer really is surveillance. Constant global surveillance. We have to watch this virus like a hawk, tracking these changes as they happen so we can try to anticipate what's coming next and get our vaccines ready. It's a never-ending battle.